Today we will discuss about a paper where they attempt to make a biomarker that can detect different kind of cancers, or we can say that universal biomarkers. And what they have find out is that this can PCNA be a universal biomarkers for cancer detections or not. So we will discuss a couple of figures that are presented in this in this paper. So first figure is about the check this PCNA in different proliferating cells, that is hex 293 cells, cos7s, HeLa cells. And what they did is they checked the presence of PCNA in the cell lysate and also in the cell culture media. And along with this PCNA, they also checked the other proliferating markers also, such as MCM2, E2F1, AURK, CDK1, TROP2, PLK1. And what they are able to see is that these markers are present in the cell lysate, but they are not present in the cell culture media, whereas PCNA is present in both the cell lysate and also in the cell culture media. So this can be a good candidate for the biomarkers, as we don't need to do the biopsy and we can directly take the fluid of the suspected patients to check whether the PCNA is present in those patient in high amount or not. And once they identified that PCNA could be a good candidate for the proliferating markers, what they did is they check this PCNA in different cell lines. That is, first one is this HEC293 cell lines, second one is this mouse that is infected with the cancer cells, and C3 is also one of the controls. And from this 1 to 30, what they check the presence of this PCNA. They check the presence of PCNA in different exosomes. They check the presence of PCNA in different cancer cell lines. And before directly moving into this figure 2, one thing that they have presented, or they have pointed out in their paper, is that this PCNA is usually found PCNA is usually found either in the multiple states, or we can say either in the dimer or trimers, but they are not found in monomers. So from that they are able to hypothesize that PCNA could be present in the extracellular vesicles. So from this and observations, they purified the extracellular these exosomes that are secreted from the cancer cells and then check the presence of PCNA. And what they are able to see is that once the exosomes are depleted from the cell culture media, the PCNA presence in the media goes down. This indicates that PCNA is secreted by the cancer cells in the form of exosomes. And once the exosomes, the cell culture media are depleted or devoid of these exosomes, then PCNA signals goes down drastically. So thereafter, once they find out that from where this PCNA is being secreted, how this PCNA is being secreted by the cancer cells, they checked this presence of PCNA in different kind of cancers that are presented here in the form of numbers. It is 1 to 30 cancers, and they are able to see that the presence of PCNA in the exosomes of these cancer cell lines, along with this hex cell lines, which is act as a control, and the C3 is the cell lines where cell culture media is devoid of exosomes, and C2 is from the mouse. There is a mouse that is infected with those proliferating cells. Here also we see some presence of the PCNA. So from these two figures, they made a hypothesis that PCNA could be a good candidate for the universal biomarkers for cancer detections without requiring for any biopsy, and can be detected directly from the fluid of the suspected patients. If you need to understand more about this approach or these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.